Hello everyone. In today's class, we'll be dealing with the remaining topics in carbohydrates and then we'll be moving on to proteins. Okay. So the first topic which we are going to deal is furanose and pyranose rings. Okay. We have already discussed the straight chain structure of monosaccharides, that is the glucose straight chain structure. What happens is this glucose, we saw that it can form ring-like structures. But ring-like structure exists in how many forms? Two forms. That is what we are going to discuss today. The furanose as well as the pyranose form of the rings. Okay. First, furanose ring. It is considered to be a 5-membered cyclic structure. You can see in this diagram a 5-membered structure. Okay. What happens is, how does it forms? The fourth carbon, the hydroxyl group at the fourth carbon will be reacting with the carbonyl group. That is, this carbonyl group could be either a aldehyde group or a keto group, which is C1 in aldoses and C2 in ketoses. And in the case of a pyranose ring, it is a 6-membered cyclic structure where again the hydroxyl group. So whenever we talk regarding the furanose and pyranose, we are talking specifically about the hydroxyl groups. Here the hydroxyl group at the C4, and in pyranose it is the hydroxyl group of the C5. Okay, it gets reacted with C1 of aldoses or C2 of ketoses. So let us understand with the help of the diagram. So as you can see in this diagram, this is a D-glucose. It is having 6 carbons. What will happen in the case of furanose is, which carbon? Fourth carbon. That is 1, 2, 3, 4. This fourth carbon's alcohol group will react with the carbonyl group and it will form a furanose ring. Why there are two structures? Because this is representing the alpha and this is representing the beta, which we discussed last class. See, in alpha the alcohol group is facing downwards and in beta isomer the alcohol is facing upwards on the same side as compared to CH2OH. But when we talk about pyranose rings, what is the condition? C5, right? So fifth carbon alcohol group will react with any of the carbonyl group. Here it is a aldose, that is why it is reacting with the aldehyde group and it forms a 6-membered ring. Again, this is an alpha form and this is a beta form. I hope you have understood. Now we'll move on to the next topic. So the next topic, we actually dealt with it, but let us also understand it with the help of a diagram. Okay, so glycosidic linkages. Are glycosidic linkages in monosaccharides or in oligosaccharides and polysaccharides? Polysaccharides as well as oligosaccharides. Sucrose between glucose and fructose. Mannose between two glucose subunits. Lactose between glucose and galactose. You have to remember everything, okay? So maltose, also known as malt sugar, which is a disaccharide. These are disaccharides. Malt sugar is composed of two glucose units. They are connected by alpha 1, 4 glycosidic bond. Why this is known as alpha 1, 4 glycosidic bond? Because I'll tell it you through a diagram. Okay, see in this structure you can locate maltose at the bottom. Now this is a glucose molecule, first molecule, right? Now if you look here, there was a earlier when they were separate, it was what group? OH group, alcohol group. This alcohol group is facing downwards or upwards? Downwards. That is why this is considered to be a alpha. Okay, so since this is an alpha and it is starting from alpha, that is why the names come. Name is, the type of the bond is alpha 1, 4. That is the first glucose, first carbon, and the second glucose, fourth carbon get connected by removal of a H2O molecule. Got it? So this is what is maltose. In the side of glucose, ரெண்டு எண்ட்லயும் குளுக்கோஸ் இருந்தா அதுதான் என்னது maltose. Okay. This we already seen the definition. Now in the case of lactose, we have galactose and glucose, which are joined by what? I've written beta 1 comma 4 glycosidic bond. Shall we see in the form of a diagram? See, so that is lactose. Here it is. See, this is a galactose molecule. Again, the OH group is facing, earlier when it was not connected, it was facing upwards. OH group மேல இருந்தா என்ன அர்த்தம்? என்ன ஐசோமர்? பீட்டா ஐசோமர் தானே. Beta isomer is at the starting point, right? Starting point of the molecule. Varshita, are you listening? So that is why this is considered to be what? Beta 1 first carbon, fourth carbon, glucose molecule. Beta 1 comma 4 glycosidic linkages. Okay. So now in the case of sucrose, the type of the bond is alpha. It is an alpha 1 comma 2. Okay. Which side is alpha? That is, sucrose is having glucose as well as fructose. Whether glucose is alpha or glucose is alpha. Okay. So see in this diagram. This is your glucose molecule. This is your fructose molecule. Fructose what? Pyranose or furanose? Pyranose. Now you saw, right? Fructose mostly exists in furanose. Sorry. This see. So fructose shows furanose, and furanose is actually the 5 carbon, right? That is the 5-membered ring. This is furanose structure. This is pyranose structure. Glucose is in pyranose structure. Furanose is in, that is fructose is in furanose structure. And this is alpha glucose. That is by alpha 1 comma 2, not 1 comma 4 in this case. 2. ஏன்னா ரெண்டாவது கார்பன் யாரோடது? fructose ஓடது. புரிஞ்சிருச்சா? Understood? Okay. This is what the structure is. You can have a note of this. Now all these structures are getting together, forming bond by removal of H2O molecules. You have to remember that. Okay. Okay, I hope you have noted. Now we'll move on to the next topic, that is forms of oligosaccharides, trisaccharide, tetrasaccharide and pentasaccharide other examples. Okay, so first we'll deal with the trisaccharide. The example given here is raffinose. What is raffinose? Trisaccharide na எத்தனை units இருக்கும்? Monosaccharide unit. 3 units. What are those 3 units? One is galactose, next glucose, next fructose. All these are connected through what type of linkages again? Glycosidic linkages. Okay. So it is this raffinose. What is the example? Raffinose. Raffinose is a form of a trisaccharide and it is seen in beans, cabbage and legumes. Okay. This is not digestible by humans due to lack of alpha galactosidase enzyme, leads to gas formation in colon. Colon means intestine. Intestine la gas formation நடந்துரும். எதனால? ஏன்னா ராஃபினோஸை நம்மளால என்ன பண்ண முடியாது, டைஜஸ்ட் பண்ண முடியாது. ஓகேவா? சரி. Next is a form of a tetrasaccharide. How many units? 4. So what they are, you can see here, one glucose, one fructose and 2 galactose. ரெண்டு galactose இருக்குங்க. மீதி எது? ஒரு glucose ஒரு fructose. Okay. Now this is also present in beans, soy products. They serve as a carbohydrate reserve in plants but indigestible in humans. Again, both raffinose as well as stachyose are considered to be indigestible. Okay. The next is a pentasaccharide. It will have how many carbon atoms? Monosaccharide 5. Galactose 3 units, remaining glucose, fructose. See, 3 galactose, 1 glucose, 1 fructose. Again found in leguminaceae and plays a role in plant energy storage, but humans again cannot hydrolyze it fully. Okay. So what you have to remember in these type of questions is whether raffinose is a trisaccharide, whether stachyose is a tetrasaccharide, whether Biomolecules is a tetrasaccharide. Okay, and their composition, pentasaccharides, and their compositions to be remembered because questions could be asked only on those particular topics. Okay, so now we'll move on to the next. Okay, so this was an easy form. Next, polysaccharides. Yesterday we classified polysaccharides into homoglycans and heteroglycans, right? We told that polysaccharides are not considered what? Not considered sugars, right? They are not sugars. They are called what? They are called glycans. Now what is considered pentosens and hexosens? What this is? Why it is ending with ANS, ANS? What is the reason? It means a polysaccharide. We know polysaccharide is made up of a repeating unit of a monosaccharide, right? If that monosaccharide is a 5-membered ring, if that monosaccharide unit within a polysaccharide chain is a 5-membered ring, then it is a pentosan polysaccharide. Pentose sugar is different. Pentose, if we say it is a monosaccharide, but if we say pentosens, it means polysaccharide. And in the case of hexosens, what it means? Each monosaccharide unit is containing 6-membered ring. That is what it clearly means. Okay. So pentosens, polysaccharides composed of pentose, 5 carbon sugars. 5 carbon sugars such as arabinose, xylose. Arabinose is a monosaccharide. Xylose is a monosaccharide. If it is containing arabinose, it is considered arabens. What? Arabens. Arabens is a polysaccharide, but arabinose is a monosaccharide. Are you getting me? And again, xylose is a mono, but many xylose together through glycosidic bonds. What they are? Silence. What? Silence. Easy concept, right? Okay. Examples are arabens, which are composed of arabinose, and the other example is silence, which is composed of xylose. Arabens are found Yesterday we discussed, right? Gums, right? Gums of trees, ramens, and xylons are formed composed in the wooden tissues. Okay, so this is a common in plant cell walls and contribute to its rigidity. Again, hexosons. What? Hexoson? Six-membered ring, six carbons. Okay, six carbon sugars. That is, the monosaccharide unit is a six carbon sugar. What it is? It could be either glucose as the unit, it could be either mannose, or it could be either galactose as the monosaccharide unit. Example: starch. What? Starch. Cellulose, glucose, that is glycogen, not glucose, and galactans. These are often found in plants as well as animals. Easy? What what is the functional significance? Pentosons are structural polysaccharides. What? They are forming the structural compositions of organisms. Whereas hexoson serves both structural as well as storage roles. Very important. Okay. Questions can be asked again in which form? Not in the form of what is pentosan, what is. Questions will be directly asked. What is a silane? Silane consists of what? What is araban? Araban consists of what? Okay, in those ways. Is it clear? Okay, you can have a note. Amingla, listen. Neat relevance is written here after completion of class. When you get this material, you can also remember from which concept the questions can be asked. Okay, for your understanding. Okay. Any doubt? Ami pentosan na etana? 4 4 இல்லவே இல்லடா டேய் 5 கார்பன் அது கேட்டேன். I hope it is completed. So we'll move on to the next topic, that is homoglycans. What? Homoglycans, also known as homopolysaccharide. What is a homopolysaccharide? Yesterday we discussed the monosaccharide units are different or of the same type. Same type of monosaccharide units. If it is having glucose, it will have glucose continuously. Okay, that is what it means. The first example which we are going to deal is cellulose. Okay, cellulose, as we already know, it is the composition of what? Cell wall, right? What type of cell wall? Primary cell, plant cell wall only. Okay, primary cell wall. Okay, cell. Now its composition and structure. Now this cellulose is composed of beta D glucose acids monosaccharide units, as you can see here. Beta D glucose acids monosaccharide units, again connected by glycosidic linkages. What type of glycosidic linkages? Beta 1,4 glycosidic bonds. Okay, sorry. Again, these forms microfibrils due to the property of hydrogen bonding. Okay, see in this diagram. So this is a cell wall. This is a cell. It is surrounded by a cell wall. This cell wall consists of பார்க்கிறது எப்படி இருக்கு? ஃபைபரா இருக்கா இந்த டயகிராம்? This is known as microfibrils. Why this microfibrillar structure is seen is, see, this is one cellulose chain, this is one cellulose chain, this is one cellulose chain. Between each chain, what there is? What these dotted lines represent? Hydrogen bonds. These are hydrogen bonds. அப்போ ஒவ்வொரு chainம் are attracted by each other, causing what type of structure? Fibrillar structure, which is known as microfibrils. In cell biology, cell the unit of life, we discussed cellulose microfibrils, right? That is what this is. Okay, so again, what type of glucose? Beta D glucose monosaccharide units connected by what? Which type of glycosidic linkage? Beta 1, 4. Okay, 1 to 4. 1 வந்து glucose, 4 வந்து பக்கத்துல இருக்கிற glucose. சரிங்களா? சரி. Molecular weight, it is exceeding several hundreds. Okay, greater than around 800 daltons. Okay. Sorry. Next, when the cellulose gets digested partially, not fully, partially digested, what that form is known as? If it gets partially digested, it is known as cellobiose. What? Cellobiose. What happens is the beta 1, 4 glycosidic bonds break and they form a intermediate structure of limited chains of glucose molecules. Those are considered to be what? Cellobiose. Again, the cellobiose degrades and forms separate glucose. Okay, I told this partial digestion occurs when, I'll tell an example. Suppose a fungal pathogen infects host leaves, plant host, fungal pathogen. Fungus, what it does is it produces enzymes. What enzymes? Cell wall degrading enzymes. Okay, what this cell wall is, cell wall component which we are discussing now is cellulose, right? So in order to break cellulose, what would be the enzyme? Cellulase. When this enzyme acts on the cellulose, what it does is it breaks which bond? 1, 4, beta 1, 4. It is alpha 1, 4 glycosidic bonds and cellobiosis form. Then cellobiosis is again broken down and it forms separate glucose molecules, which can be used as a nutritive form for what? For the, see how he is getting benefit. Okay, sorry, this is understood. Okay, occurrence, wood, the wood, 50% of the wood contains cellulose, and cotton, 90% of the cotton contains cellulose. Okay, the most cellulose is the most abundant organic molecule. Organic molecule means again it contains carbon and it is the composition of a living system. Okay, very important. So again, a very important point which might be asked in our question paper. Okay, tunicin. Tunicin is found in urochordates. Okay, it is not a plant; it is a invertebrate, as we know. Intermediate. Sorry. So it consists of a cellulose-like of structure. That is what is tunicin. Which organism? Urochordates. It is termed as animal cellulose. Okay, tunicin, which is like cellulose, is considered to be a animal form of a cellulose. Okay, so it is a rare example of cellulose in animals. When we tell cellulose, it means plants, but even animals of this kind consist of cellulose-like structures. Okay, which is known as tunicin. Okay, so you can have a note of this. The type of linkage is beta 1 comma 4 glycosidic bonds. Why? Because the glucose molecule which is present as the monosaccharide is not alpha D glucose, it is beta D glucose in cellulose. Okay, you have to remember this. Okay, now we'll move on to the next topic. Another form of a homopolysaccharide, which is nothing but starch. Okay, this is the diagram for cellulose. We have already dealt with it. Next is starch. We have seen that starch is seen in which potatoes, right? Potatoes contain what form of polysaccharide is starch. Okay, so starch is composed of what type of glucose? Alpha D glucose. Cellulose la beta D glucose. Starch la alpha D glucose, and this alpha D glucose chain vana form panna podhu. It is going to form a chain, right? Now this chain in starch is of two forms. What I'm saying, alpha D glucose, two types of chains. One is considered to be amylose, the other is amylopectin. Amylose, unbranched structure, unbranched chain. Amylopectin, branched chain. Okay, got it? Now we'll understand here. See, so it consists of two forms, that is, one is amylose and the other is amylopectin. Within a starch molecule, most of the type of chain is amylopectin. Sir, how much, sir? What is the percentage? 80% of the starch is composed of amylopectin. The remaining 20% is amylose. This is linear chain, which is connected by what type of bond? Alpha 1 comma 4 bond only, glycosidic bond. Okay, but amylopectin is connected by two types of bond. If it is the same straight chain, it is alpha 1 comma 4 bond only. If a separate branch is coming, then it is alpha 1 comma 6 glycosidic bond. You have to remember, very easy. Okay, so alpha 1 comma 4, alpha 1 comma 6. Now, this amylose consists of about 250 to 300 glucose units, whereas amylopectin consists of 30 glucose units. I'll tell it again. Amylose consists of 250 to 300 glucose units, alpha D glucose units, whereas amylopectin consists of about 30 glucose units only. Okay, so we'll understand it in the form of a diagram. See, this is a straight chain, and what this is? Amylose or amylopectin? Amylose. What are these bonds? Alpha 1 comma 4. Again, this is amylopectin. Okay, in the mari branches follow. Any branches come here. Now the straight chain is having what bond? 1 comma 4. If a branch is coming, it forms what bond? 1 comma 6. What it means? This molecule, from this molecule, which carbon? Sixth carbon. And from the above molecule, first carbon. That's it. Okay. So alpha, this is alpha D glucose, this is also alpha D glucose. That is why alpha 1 comma 6, 1 comma 4. Okay. Whenever we write the name, we go from right to left. That is why this name is in that way. Okay. Understood? Sit. Now what are other important point is amylose is helical, less soluble. Usually we know that polysaccharides are insoluble, but some forms are soluble. Okay, this is less soluble. Amylopectin is highly branched and more soluble. Okay, now the other important point to note here is the reaction with iodine. Reaction with iodine. Iodine is used in starch test, right? What are those appearances which can be seen? Violet, blue, red. Violet, blue, red. When will violet be produced? See, amylose gives a deep blue color. Amylose, which is a straight chain, will give a blue color when reacted with iodine. Whereas amylopectin will give a red color or reddish brown color. Okay. What if you take the whole starch molecule, not separately, as amylose, not separately as amylopectin, together in the form of potato tissues? Potato tissue la iodine ஊத்துனா என்ன color கொடுக்கும்? Violet. In such a case, in the case of a whole molecule starch, it forms violet color. Okay, got it? Understood? So for amylose, it is blue color. For amylopectin, which is branched, is red color. Okay, sorry. Starch is the primary storage carbohydrate in plants. Potato starch is a classical example containing both amylose as well as amylopectin in these properties. Okay, in these proportions. Okay, sorry. Now you can have a note of these things. There are many points in this particular topic, that is starch. What are those important points? Amylose, amylopectin, reaction with iodine, type of glycosidic bond. Amylopectin, one more type of glycosidic bond. Together as starch, they give different type of color. Questions can be asked from any nook and corner. Okay. We have seen the structure of starch, right? Amylopectin. You need me to show the diagram? This completed? Completed. Then I'll. Okay. Okay, so I'll show you the diagram. So this is the diagram. Okay, mainly mainly this diagram seems to be different. This is easy, 1 comma 4 glycosidic bond, but here the only difference is this is the branch. When it arises, it forms a alpha 1 comma 6 glycosidic bond. Okay, so now we'll move on to the next topic. Okay, so next type of the homopolysaccharide is glycogen. Okay, it is made up of again alpha D glucose units and is connected again with alpha 1 comma 4 linkages. What is the difference is it is also containing alpha 1 comma 6 bonds only in the form of branches, but the branches are more. Glycogen is similar to starch only, but it is highly branched. That is what the difference is. This is a more highly branched than amylopectin. It is specifically when you compare it with amylopectin, it is highly branched. Okay, it, every branch every branch consists of about 8 to 12 glucose units in glycogen. Okay, now starch was a component of what? Plants. Glycogen is seen in animal cells. Okay, like stored mainly in liver to 10% of the fresh weight, muscles 1 to 2%, and it, it acts as a glucose reserve in animals. Okay, in animals, செல்லுக்குள்ள glucose reserve இது வந்து ஆக்ட் பண்ணும் glycogen. ஓகேவா? சரி. Properties: it is soluble, quickly mobilized by glycogen phosphorylase enzyme, provides glucose during fasting. When we take up fasting during religious festivals or intense muscular activity. Okay, easy. Sorry. Iodine test. What will happen here? Again, glycogen as a whole will give reddish brown color. Why? Earlier, where we saw reddish brown, amylopectin. It is similar to amylopectin. That is, it is little bit highly branched. Okay, that is where the same color is given in iodine test. Okay, so this is what is your glycogen. Okay, now we'll move on to the next topic. That is chitin. Okay, chitin we have discussed, but structurally what it contains needed to be dealt with. Remember, in when we discussed in cell the unit of life, we saw cell walls of different organisms. N-acetyl D-glucosamine. We discussed, right? NAG. NAG is the component of what? Chitin. Glucosamine also, previous class we discussed. Derived form of a monosaccharide, amino sugar, right? So, glucosamine, what glucosamine? N-acetyl D-glucosamine, okay, D form. Sorry. Linked by beta 1 comma 4 bonds. Chitin is, what is the monosaccharide unit here? N-acetyl D-glucosamine connected by beta 1 comma 4 glycosidic bonds. Okay, structurally it is similar to cellulose, but with acetylated what? Amino groups NH2. Okay, அதனால தான் நம்ம அதை என்னன்னு சொல்றோம்? Glucosamine அப்படின்னு சொல்றோம். Where it is found? We discussed it in the last class. In the exoskeleton of arthropods, example cockroaches, crustaceans, etc. And also in the fungal cell walls. Okay, rigidity and protection. Tough, insoluble. Starch, some part remains soluble. Here, insoluble because it is the cell wall material. Okay, that is why. Resistant to many chemicals, stems by combining with proteins and calcium carbonate. Okay, in crustaceans, they have calcium carbonate along with chitin. Okay, so this was your chitin. Now we'll move on to the next, that is heteropolysaccharide. Heteropolysaccharide means the monosaccharide units are same or different. Monosaccharide units are different keyword. Okay, how different? Let us Deal with the example. What example? Hyaluronic acid. Hyaluronic acid is composed of what you know? It is composed of disaccharide units like D-glucuronic acid and N-acetyl D-glucosamine. First, glucuronic acid. Next, N-acetyl D-glucosamine. They are alternatively arranged in the form of a chain. That is what is hyaluronic acid. Connected by what type of bonds? Beta 1, 3 and beta 1, 4 bonds. Okay. Where they are found is they are found in the synovial fluids between the joints as well as vitreous humor of the eye as well as skin, connective tissues, especially in umbilical cord. This is important. This will be asked in the questions. Okay, sorry. And part of the extracellular matrix. Okay. Now properties: hydrophilic. Hydrophilic means attracts or repels. Attracts water. Okay. Sorry. Viscous in nature. Large amounts of water provides lubrication, elasticity, and shock absorption. Act as animal cement. Very important. Animal cement. Where? On the cell coats of animal cells. Okay. Which? Hyaluronic acid. Okay. The next heteropolysaccharide is chondroitin. Okay. Chondroitin. Okay. Sorry. It is found in the cartilage. Now it is composed of, earlier we saw glucosamine, right? N-acetyl glucosamine. Here the difference is galactosamine. But the first unit is the same. D-glucuronic acid. Second unit is different here. Galactosamine. Okay, wow. Now again this is connected by beta 1, 3 and beta 1, 4. Structurally, only one similarity, that the first unit of both hyaluronic acid and this particular thing is same, but the second unit is different. But people tend to confuse and they think that both are structurally same. Moreover, this D galactosamine is sulfate. It has SO3 groups attached to SO4 groups attached to it. Okay, sulfated compound found abundantly in cartilage, tendons, as well as in ligaments. Contribute to tensile strength. You know what is tendon and ligaments, right? Connect bones to muscles and ligaments bones to bones. Okay, so they are highly negatively charged, have sulfate groups. Sulfate group is again what in nature? Negative or positive? Negative in nature. They attract water. Water being polar in nature. Phosphate is again, that is sulfate is again negative in nature. Polar and polar things get attracted. And provides resilience to cartilage by resisting compression. Easy? Okay. Now the next heteropolysaccharide here is heparin. Okay. It is anticoagulant of blood. What is anticoagulant? Anticoagulant means avoids blood clotting. Okay, prevents blood clotting. Okay, do you know what is the enzyme of blood clotting which is meant for blood clotting? Any idea? Thrombin. Thrombin. Okay, thrombin is the enzyme which is responsible for blood clotting. What then? What does heparin do? It will inhibit this enzyme. That's it. It will inhibit this enzyme, so blood clotting will not happen. Okay, so polymer of sulfated disaccharides containing glucuronic acid or iduronic acid and glucosamine residues, highly sulfated, that is by a negative charge. Okay, produced by mast cells. Mast cells are nothing but WBCs. Okay, they are found in liver, lungs, blood vessel walls. Okay, so strongly acidic, highly sulfated, and one of the most negatively charged biomolecules. One of the most negatively charged biomolecules, and binds antithrombin 3. This is a protein. What this heparin does is It binds with a protein called antithrombin 3. When it binds with it, it regulates it, it activates it. Which protein? This protein, antithrombin 3. When this antithrombin 3 protein gets activated, it inhibits thrombin. Eventually, blood clotting is prevented. Got it? Sorry. Okay? Sorry. So, this was an important thing. Then pectin. Where did we discuss pectin in cell, the unit of life? Middle lamella, cell wall material, middle lamella, calcium and magnesium pectates. What is pectate or pectin? Pectin is again a heteropolysaccharide composed of galacturonic acid with rhamnose. Yesterday we discussed rhamnose, right? Huh? Hemicellulose component which lacks hydrogen atom yesterday. That not raffinose. Raffinose we discussed that. Rhamnose yesterday's class. Okay. So rhamnose, arabinose, galactose all combined to form the middle lamella. Okay. Sorry. Abundant in the middle lamella, primary cell walls of plants, especially in fruits. They are considered to be cements of cells. That is why they contribute to cell adhesion. Okay. They are hydrophilic in nature, gel forming when combined with water, and used in food industry for making jellies as well as jams. Okay. So pectin is an important thing. Questions are repeatedly asked in pectin from the cell unit of life chapter itself, right? It is composed of calcium and magnesium pectins. But as per NCRT, calcium pectins. Okay. Sorry. Now the other is hemicellulose. Hemicellulose is a matrix polysaccharide in the plant cell walls. Now this is composed of xylose. Xylose is a what? Monosaccharide or polysaccharide? Xylene, not xylose. Monosaccharide. This is one type. Then arabinose, different type. Then again mannose, different. Galactose, different. Glucose, different. All are same or different? Different. That is why this is known as heteropolysaccharide. Distinct from cellulose in structure and solubility. Okay. Occurrence found in plant cells walls associated with cellulose microfibers, which we just saw. Also stored in special plants like, important point, phytolabus, ivory palm, okay, where hemicellulose act as a reserve carbohydrate. Okay. Now this phytolabus, phytolabus, hemicellulose commercially act as vegetable ivory and used in producing artificial ivories. Okay. From which organism? Phytolabus. Phytolabus. Okay. So this is what hemicellulose is all about. Okay. By this we end heteropolysaccharide. Now one thing in polysaccharide is left, that is mucopolysaccharides. Now this has nothing to do with the earlier thing, that is homo, hetero, nothing. Separate classification. Okay. What are mucopolysaccharides are? Slimy polysaccharides, which are slimy in nature. Okay. And they consist of glycosaminoglycans. Okay. Long unbranched heteropolysaccharides. See, hetero. Okay. Repeating disaccharides with amino sugars and uronic acid. Most important point here is they bind water. They bind water molecule with proteins. Mucopolysaccharides are polysaccharides which bind water and protein. Okay. Now types, hyaluronic, that is those things which we earlier saw, right? Those types only. Chondroitin sulfate, heparin, dermatan sulfate, keratin sulfate. Each varies in sugar reduce and sulfation. Okay. Now properties, again they are highly negatively charged due to the sulfation. Okay. Hydrophilic, binding large amounts of water with protein. Okay. Form complexes with protein to create hydrated gels in extracellular matrix. Okay. This is what the important point regarding this is. involved in lubrication as we saw earlier. Okay, not, you have to just remember one thing: slimy polysaccharide binds water with proteins, consists of glycosaminoglycans. Okay, that's it. Okay, now very important thing which we missed last: composition of protoplasm. Protoplasm means what? Protoplasm na ana tambi? Hmm? Cytoplasm or protoplasm or same or different? Different. Why? Cytoplasm plus plus nucleus. If we not combine, if we consider them together, it is known as protoplasm. Cytoplasm is the fluid matrix which is containing all the organelles. Okay, and if we also consider nucleus to be the part of it, then it is considered to be the protoplasm. In the protoplasm of the cell, any form of a cell is mostly composed of water. Okay, 70 to 90% of every cell is composed of water. Okay, next major compound, any idea? Protein. About 15 to 16% if you consider a separate cell. Here they are written 60 to 70% when considered in the form of a dry weight. What is dry weight? You take a cell or a tissue, you remove the moisture. Water is removed now, which was earlier the most abundant thing. Now at that point only it is considered to be 60 to 70% in the case of animal cells. In the case of plant cells, the dry weight is maximum by carbohydrates. Okay, about again 60 to 70, 80%. Okay, sorry. Now lipids 1 to 3%, carbohydrate 1 to 2% of protoplasm, and not in the form of dry weight. Normally, okay, normally the cell is consisting of 1 to 2% of the protoplasm and mostly abundant in the plant cells. Okay, inorganic compound like salts, acid, bases, gases, these are about 1 to 3%. Okay, so you have to remember two things. Most abundant thing is water. How much? 70 to 90%. Second abundant is protein. Protein is 15 to 16%. Okay, in animals, dry weight is mostly composed by proteins. In plant cells, dry weight is mostly composed by carbohydrates. Okay, got it? Now we'll move on to the summary is not required. Some practice MCQs. Which of the following disaccharide is non-reducing? Maltose, lactose, sucrose, cellulose. Same question we discussed last class, right? What is the answer? Same question தான் கேட்டிருக்கேன். If you are not answering, then reducing sugar. Which? Maltose. Why are you telling maltose? Maltose is a disaccharide. Lactose is also disaccharide. Cellulose, it is partial digested form of cellulose. Sucrose, disaccharide. Then what is the answer is? Answer is sucrose. Sucrose was in the option. Okay, so sucrose is considered to be a non-reducing sugar because both anomeric, that is first carbon, are involved in glycosidic bonds. Okay, so sucrose is what again? Reducing, even non-reducing, even though it is a disaccharide. Okay, which linkage is present in cellulose? Cellulose, कितना linkage है? Alpha 1, 4, beta 1, 4, beta 1, 4, beta 1, 6, alpha 1, 6, not right. So the answer to this is again beta 1, 4 linkage. Okay. The next question: starch contains amylose and amylopectin in which composition? Amylose 20, amylopectin. So this is in the form of a pair. So the answer is which one? C. C or D? The question seems to be not proper. B. Okay. Sorry. Which heteropolysaccharide is called animal cement? Animal cement. It is told right. Heparin, chondroitin, hyaluronic acid, pectin. Pectin, plant. Hyaluronic acid, heparin. What is the answer? Hyaluronic acid. Saringla? Sorry. In animal cells, the most abundant component of dry weight is animal cell protein. Okay. So since the last question was for protein, now we'll be moving on to the next topic, that is proteins. Okay. Proteins. Now we'll deal with proteins. Carbohydrates khatam, over. Now we are dealing with proteins. Now only proteins. So again, most abundant organic molecule in living system. Molecule, as a molecule, not as a compound. Compound we saw, right? Different. Okay. So they form about 5% or 50% of the dry weight. Okay. Most important point here is when protein as a long chain of a monomer. I'll not reveal it what the monomer. You might know it, but upon hydrolysis of that long chain, what is produced is amino acid. Then what is considered the monomer of protein? Amino acid. Proteins are nothing but chain of amino acids. What is amino acid which is having both amine group and an acid group? We'll be dealing with it. Okay. So then the next, there are about 300 amino acids, but not all amino acids are involved in the protein formation. Mukyama 20 amino acids. 20 standard amino acids are only used in protein formation. Okay. Sorry. Collagen is the most abundant animal protein, and plant rubisco enzyme. Okay. Ribulose biphosphate carboxylase or oxygenase. Right. Full form I am saying. Okay. Protein synthesis is controlled by DNA, central dogma of molecular biology. Okay. Understood? Sir. Next, amino acid. We'll see the structure. The basic unit of protein is amino acid, right? So amino acid is important. Center or carbon, center one carbon, 4 sides, different functional group. Not even one functional group is same. All are different. What type of carbon then? If you frame a mirror image, it will be exactly opposite. It will not be symmetrical, it will be asymmetrical. Asymmetrical carbons are considered what carbons? We're taking so much time. Yesterday also we discussed, right? Alpha only, alpha carbon only. That is alpha. Alpha is considered because this is the carbon which is beside it. Chiral carbon. Chiral carbon. Okay. So this is considered to be the chiral carbon. Why? ஏன்னா அதை சுத்தி carboxylic acid group இருக்கு, amino group, this is NH2 group, H group இருக்கு, hydrogen atom இருக்கு, or side chain. This side chain can also be long enough. Okay, it could be alkyl or aryl, both aliphatic, aromatic, both form. Okay, so since it is having 4 types of functional group, it is considered to be a chiral compound. So amino acid what? Chiral compound. It will undergo what? Optical activity. Sorry. It has amino group and acid group. Amino group irukku, acid group irukku. அதனால தான் என்னன்னு சொல்றாங்க? Amino acid ன்னு சொல்றாங்க. Amino acid தான் சொல்றாங்க. Easy. NH2 group is basic. NH2 group ஓட pH என்னது? Basic pH ஆ இருக்கு. Alkaline in nature. COH group பேரே என்னது? Acid. அப்ப acidic in nature. சரிங்களா? But this compound also exists in which form? Zwitter ion, that is the alpha carbon. The alpha carbon will have H or here COO minus or plus? Minus. This side NH 4 or 2 here H removed and came here. What? Plus NH4 plus. In the side plus, in the side minus. ரெண்டுமே இருக்கா? Cation இருக்கு, anion இருக்கு. Both are in the same structure. அந்த மாதிரி ஸ்ட்ரக்சரை தான் நம்ம என்னன்னு சொல்லுவோம்? Zwitter ion. Okay. Zwitter ion இருக்கிற இந்த பாயிண்ட் என்ன பாயிண்ட்னு சொல்லுவாங்கன்னா, what point? Iso electric point or pH. It's electric point or pH. In this condition, whether it is acid or base, neutral, not acid, not base, it is considered to be neutral. Neutral oda pH எவ்வளவு தம்பி? 7. கரெக்டா? ஈஸியா இது? அப்போ NH4 plus ஆவும் இருக்கு, CO minus ஆவும் இருக்கு ஒரே ஸ்ட்ரக்சர்ல. Then it is considered what? Zwitter ion. Point is known as isoelectric point or pH. Okay. Now you tell me what this structure is. இப்போ இந்த alpha amino acid இருக்குல்ல, whether this is acidic or basic? Basic in nature. Yeah. This end. Okay. Basic acidic? Okay. Basic in nature. சரிங்களா? Okay. So this is what the structure and the Zwitter ion and the concept is all about. Now we'll deal in a specific condition. We told that alpha amino acid are considered to be chiral in nature, except one. Except one amino acid is not chiral. For chirality, all the 4 functional groups should be different. But what if a functional group is repeated? Whether it will be chiral? That is what the example is. The example is which amino acid? Glycine. Glycine is having no R group. Side chain illa. The side chain badla what it is again? H group. Okay. Okay. What this is? Amino group, acid group. What this is? Hydrogen. Ingyo hydrogen hai, ingo hydrogen. Repeat agada. Repeat agna chiral nu sollama. Chiral illa. Appo glycine is not chiral. Questions previous year question paper la ketirukanga. They are asked. Okay. Got it? Easy? Any doubt? That is in the form of a ion. 3 plus, sorry. NH. Okay, we'll deal with it. Not a very big deal. Okay, so all amino acids contain carbon, hydrogen, and oxygen. M, but some of them additionally contains sulfur also. Okay, physical and chemical properties of amino acids are due to the amino and carboxylic group and the R side chain which is present. Okay, now amino acids are differentiated into 7 groups. They are considered to be classified into 7 groups. What those 7 groups are? Neutral, acidic, basic, yes containing, alcoholic, aromatic, heteroglycic. Before we deal with this, what you guys can do is you can have a note of proteins. Note it down. Put the heading as proteins, not the same, okay? Are you able to understand whatever I am saying? Any doubts? It is not NH4 plus, it is NH3 plus. Okay, not 4 plus, 3 plus. I've checked it now. Okay, because it is already NH2. If one hydrogen is coming from H, it will become 3. Is it done? Next slide. Also write about bitter ion. Okay. If you see in the third point, if R is H, it is glycine. If that R is a methyl group, that is CH3, it is considered alanine. Okay, and hydroxymethyl CH3OH, CH2OH, not CH3, CH2OH is serine. Okay, in others it can be hydrocarbon chain or a cyclic group. It could be a cyclic group, it could also be a aromatic group. Okay, so again 7 groups of amino acids. The first group, as you can see here, is neutral amino acid. What? Neutral. What we are talking about? The pH. Okay. So amino acids with one amino and one carboxyl group. Examples are glycine, alanine, valine, leucine, isoleucine. Okay. All these things. In the case of acidic, one extra carboxyl group. Acidic will have one extra carboxylic group. It is aspartic acid, asparagine, glutamic acid, glutamine. Okay, you have to memorize this because questions are often asked from here. Okay, basic one extra amino group, arginine, lysine. Okay, sulfur containing will have sulfur, cysteine, methionine. Easy, only two. Sulfur containing cysteine, methionine. Instead of R, H group, glycine. In the place of R, methyl group. In the place of R, CH2OH, serine. Okay, and sulfur containing cysteine. Okay, sorry. Alcoholic amino acids have alcohol group, CH2OH, right? Serine, threonine, tyrosine. Okay. Now aromatic questions were asked. Aromatic have cyclic structure, aromatic benzene structure, phenylalanine, tryptophan. This you have to pakka remember. Aromatic, phenylalanine, phenyl, name itself phenyl group is coming. Okay. Then tryptophan. Okay. So heterocyclic, not homocyclic, heterocyclic amino acids having N. Heterocyclic, homocyclic means homocyclic is only carbon and hydrogen in the ring. Carbon or hydrogen மட்டும் இருந்தா அது homocyclic. அதை தவிர மீதி ஏதாவது ஒரு element, nitrogen, sulfur, any those type of element are coming in the ring, heterocyclic in organic chemistry. Now if N comes, it is known as heterocyclic. Nitrogen containing are histidine, proline. Sulfur containing alcohol, serine. Okay. Aromatic, phenylalanine, tryptophan. Nitrogen containing histidine, proline. Okay, sorry. So this is the structure of the neutral alanine. This is the structure of aspartic acid, lysine, serine, alcoholic. This is sulfur. Okay, sulfur. And we have to correct this Twitter ion diagram. This is not 4. This is what? 3 plus. Okay. Here also it is 3 plus. Okay. Same. Now types of amino acids, 7 groups of amino acid, clear, right? Now we'll move on to peptide formation. Another peptide formation. Peptide na the type of bond present in between what? Successive amino acids. ரெண்டு அமினோ ஆசிடுக்கு நடுவுல இருக்கிற பாண்ட் என்னன்னு சொல்லுவாங்க? பெப்டைட் பாண்ட் சொல்லுவாங்க. கிளைகோசிடிக் பாண்ட்னா எங்க இருக்கும்? Polysaccharide and disaccharide. For proteins, we have successive amino acid contains peptide bond. How it is formed? If you see clearly here, COO minus. Here it is NH3 plus. NH3 plus. Okay. Now, from this NH3 plus, two hydrogen molecules, two hydrogen atoms, and from the CO molecule, one oxygen atom. Together what? H2O molecule, right? H2O molecule release. Glycosidic also H2O molecule, here also H2O molecule released between CO group of one of the amino acid and NH3 plus of the other amino acid. அதுல ஒரு H2O molecule போயிடுச்சுன்னா நடுவுல என்ன ஃபார்ம் ஆகும்? COONH. என்ன ஃபார்ம் ஆகும்? C double bond O N H ஃபார்ம் ஆகும். இந்த சைடு இப்படி இருக்கும், இந்த சைடு இப்படி இருக்கும். இதுதான் என்னது? What is this? என்ன bond இது? Peptide bond. Okay. Now the peptide bond is connecting. Based on how many units it is connecting, it is again categorized into dipeptide, tripeptide, polypeptide chain. Okay. So if you see chain having two amino acids linked by one peptide bond is considered to be a dipeptide. நடுவுல ஒரு peptide bond இருக்கு, பக்கத்துல ரெண்டு அமினோ ஆசிட் இருக்கு. டிஃப்ரெண்டா கூட இருக்கலாம், சேமா கூட இருக்கலாம். அதுதான் நம்ம என்னன்னு சொல்லுவோம்? டைபெப்டைடுன்னு சொல்லுவோம். சரிங்களா? ஆனா ஒரே பெப்டைட் பாண்ட் தான், ரெண்டு பெப்டைட் பாண்ட் இல்ல. சரிங்களா? The sequence of amino acid present in the polypeptide is specific for a particular protein, particular sequence. Different protein, different sequence. If the sequence is different, it is different protein. Okay, got it? Peptide bond na enadhu? CONH, formed by the removal of H2O molecule. ரெண்டு H atoms from NH3 plus, ஒரு O atom from COO minus. Okay, set. Structure of protein: primary structure, secondary structure, tertiary structure, quaternary structure. Four levels of structure. Okay. Primary structure na enaduna அப்படியே linear chain. Linear chain of the peptides, polypeptide chain is known as your primary structure. As you can see here, இது ஒரே லைன் மாதிரி இருக்கா? ஸ்ட்ரைட் லைன் இல்ல, பட் அப்படியே தனியா போகுதுல, separately it is seen, right? That is what is known as a primary structure. Refers to the number and linear sequence of amino acids in the polypeptide chain, and they have a disulfide bridges. We'll be dealing with it. Okay. The primary structure is responsible for the function of the protein. Function of the protein, what is responsible? Primary structure. Okay. The N terminal of the amino acid is written on the left side, whereas the C terminal is written on the right side. Left side what? N terminal, NH, here COO minus. Okay, right side. Is it right? That is according to you. Okay, sir. So secondary structure, what happens is folding occurs. Okay, in two ways. One is the alpha helix and the other is the beta. Okay, beta sheets. Specific coiled structures. So the folding causes a coiling. Okay, coiling effect. Three types: alpha helix, beta plated sheet, and collagen helix. Sometimes collagen helix is also considered to be the tertiary structure. Okay. Okay. Now, as you can see here, they will be forming some attractions between the N terminal and the CO terminal, causing it to fold in a way. Okay. These are considered to be what? The secondary structures. Then we'll be dealing with the tertiary structure. The helical polypeptide may fold upon itself and assume a complex but specific form. It is either a spherical form or a rod-like form or something in between, any intermediate structure. First fold ஆச்சு, ஸ்பைரலா fold ஆனா alpha helix, sheets மாதிரி fold ஆனா beta sheets, அதுக்கு மேலேயும் fold ஆகி ஸ்பெரிக்கலாவோ இல்லை டிஃப்ரெண்ட் ஃபார்ம்ஸ்ல அது ஃபார்ம் ஆனா அதுதான் என்னது? tertiary structure. இங்க பாத்தீங்களா tertiary structure? Okay. Now quaternary structure na, if they have two or more polypeptide chains, different polypeptide chains club together. Okay, that is what is quaternary. Quaternary ல என்ன ஆச்சு, ரெண்டு தனியா இருக்கிற polypeptide chain come together. For example, in which molecule? Hemoglobin, right? Hemoglobin is a protein, right, which carries oxygen. Quaternary structure. A globular protein, it is known as globular protein. We'll be discussing. So primary structure straight இருக்கும், folded structure alpha helix, beta sheets. This is tertiary structure. Any formation here, color is different. It means not the same type of polypeptide chain, different type of polypeptide chain come together. Okay, got it? Now classification of proteins. What? Classification of proteins. The classification is also classified based on different bases. Classification based on function, classification based on chemical nature and solubility, classification based on nutritional importance. Here we will be discussing based on function and structure. Okay, function and derivation. So based on the functional classification, it is classified into 1, 2, 3, 4, 5, 6, 7, 8 and 9 groups. Okay, the first is your structural protein. Gives the structure. Hair, keratin protein, keratin, collagen. Okay, now enzymatic protein, pepsin, gastric juice, right? Involved in the breaking down of peptide bonds. Okay, next transport proteins, helps to transport any form of a material. For example, hemoglobin carries oxygen. Okay, now hormonal proteins, chemical substance which also consists of some protein. Okay, for example, insulin hormone, growth hormone, these are what proteins? Hormonal proteins. Next, contractile proteins. Now in this case you have to memorize things. Okay, actin, myosin, storage protein, ova albumin, that is the albumin which is found in the egg. Okay, now genetic proteins, nucleoproteins. Okay, the proteins which are associated with what? The DNA, nucleoproteins, histones, nucleosomes we have seen, right? Defense proteins, again act in the defense. Immunoglobulins, receptor proteins. Receptor means they take up some signals, they attain signals and they react based on it. For hormones, those receptors, and for detecting the virus. Okay, so these are based on the function categorization of proteins. Now classification based on chemical nature: simple protein, conjugated protein, derived protein. Simple, composed of only amino acids, residues. Conjugated, along with amino acid there is a non-protein prosthetic group. It could be a carbohydrate group, anything. So another, this is conjugated protein. Along with amino acid there is a non-prosthetic group. Okay, now non-protein prosthetic group, derived protein, they are denatured and degraded products of the above two. If these two, simple and conjugated products, get denatured or degraded, they form this derived form of proteins. Okay, simple based on the shape or structure: globular, albumin, globulin, histones. Fibrous in structure: collagen, elastin, keratin. Okay, conjugated means what? Along with amino acid there is something else. Nucleoprotein along with the nucleic acid, DNA or RNA, proteins associated with. Glycoprotein along with glucose. Mucoprotein, mucopolysaccharides. Lipoprotein, lipids. Phosphogroups, chromo, metallic group, whatever. This is pigment. Okay. Now derived coagulated protein, proteins, metaprotein. This is just the form of a classification. Classification is important, but examples are more important. Okay, the examples which we had discussed earlier. Okay, you have to memorize everything. So that's it. We have come to the end of the session. Today we dealt with what? We dealt with proteins as well as carbohydrates. In the next class, we'll be dealing with lipids and we'll deal with this uh, folding, that is primary, secondary, and tertiary and quaternary in much more detailed thing. Okay. Okay. So thank you. Hmm.